Good evening to one and all. I'm talking about endotoxins. Endotoxins means it is one of the component which is present in a cell wall of the gram-negative bacteria, and it is typically endotoxin synonymally. It is made up of liposaccharides, lipopolysaccharides, and endotoxins are not LPS. The main function is to protect bacterial, structural, and it will be protective. So next slide. And it is large molecules coming from bacteria, and it is composed of lipids and sugars, and the toxin strength varies according to the type of the different types are there. According to their types, the toxic strength will be, and they are resistant to the heat above 100 degrees Celsius. Sir, back slide. Next, sir. Yes, this is a LPS structure where it is made up of lipid A, core, and polysaccharides. Three compartments are there. In lipid A, it is made up of diglucosamine containing fatty acids chain of 10 to 20 carbon atoms, and then core is completely made up of sugar acid residues, 2 keto and 3 deoxy dimano ketolosonic acid, that is KDO, that is polysaccharide O, that is made up of repetitive branch of the linear sugars. And also for humans and animals, endotoxin are a type of pyrogenic which induces different biological reactions. Sir, endotoxin causes, that means if the endotoxin is present means it will causes fever, tachycardia, and increases in a cardiac output, that is pain, and decreases in a peripheral resistance. And the mode of actions, how it will be action means, how the endotoxins are action means via immune system, it will be act on an immune system and the liberation of cytokines which is present in our immune system. And the sensitiveness is the genetically determined. It is genetically determined between one and another person. And also man is extremely sensitive to that few organisms which is sufficient to produce a state of shock according to their emotional shocks or any of other shocks that will be produced and that will be induced in the body. So you can see the differences between endotoxins, that is, and exotoxins. Exotoxins means that is produced in a gram positive bacteria. Endonegative is produced chiefly in gram negative bacteria, and exotoxins are highly toxic and endotoxins are weakly toxic. Then compare it to exotoxins, and it is chemically protein, and it is chemically lipid proteins contain of LPS, and exotoxins are highly antigenic. These endotoxins are weakly antigen. They are heat stable. Exotoxins will not kill in a very high heat also, but endotoxins are heat liable. This for exotoxins, vaccines are available. For endotoxins, no vaccines are available. For exotoxins, it is highly immunogenic. Endotoxins are pure immunogenic. Some of the tests are there to test the bacterial endotoxins which is present. So to detect and qualify endotoxins, this bacterial origin in an amoebocyte lysate from the horseshoe crab. It will be firstly identified in a horseshoe crab of amoebocytic lysate, and sterilization does not remove any endotoxins as they are heat stable. So FDA and USP guidelines require final product testings. We cannot easily tell that FDA and USP guidelines required, and then to safeguard against diminished effectiveness of the product due to endotoxin. So this is how to determination of the maximum valid dilution. How much dilution is there on how the endotoxins will be present, how much limited it will be. It will be used to identify by the formula, that is endotoxin limit into product concentration, how much concentration product it is, and lysate sensitivity, either it is sensitivity or not. By their lysate sensitivity we can identify. Next, sir. Yes, some of the techniques are there. One is gel cloth technique, that is by the gel information we can take it as, and chromogenic. Chromogenic means color formation of cleavage of synthetic peptide chromogenic complexes. By using the color formation we can identify the endotoxins. And also turbidimetric. Turbidimetric means by turbidity formation of the, by turbidity formation some of the cleavages will be present in an endogenous substrate. Some of the gel cloth tests are there, that is limit size, preparatory test, and quantitative test and semi quantitative tests. Gel cloth technique is one of the technique to detecting and quantifying the endotoxin based on clotting of lysate reagent in the test tubes in the presence of an endotoxin, and it will be used in a minimum concentration to cause lysate to clot under the standard conditions. So it will be used where the clotting of lysate reagent in the test tube, presence of endotoxin, if it is present, endotoxin is present, then the lysate reagents will be clotted in the test tubes, and that can be identified that sensitivity of that lysate reactivity. And also it is used to precision and validity of the test to perform the test for confirming the label lysate sensitivity and interfering techniques. This is what preparatory testing of, it is one type of the gel clot technique where this is a mechanism of action where endotoxin will be completely that is for a factor C and factor B that is used for pre-clotting enzymes or will be present, and along with 1,3-BD glucon will be present. And not this one, sir. Next slide. This is turbidometric technique. We have different types of the turbidometric technique, that is chromogenic technique and turbidometric technique. In turbidometric technique, we have endpoint turbidometric assay and kinetic turbidometric assay. In chromatogenic technique also, endpoint chromogenic assay and kinetic chromogenic assay is present. This is a turbidometric assay where it is endpoint turbidometric assay based on quantitative relationship between the concentration of endotoxins, and it will be used and turbidity of the reaction mixture after the incubation period. And also this kinetic turbidometric assay is used to measure time needed to reach for the predetermined observance and transmission of a reaction mixture. So developed in the test is carried. Measures the chromophore released for the suitable chromogenic peptide by the reaction of the endotoxin lysate. The endpoint of the chromogenic assay is based on quantitative relationship between concentration of the endotoxin and release of the chromophore. Then kinetic chromogenic assay is to measure the time and needed to reach the predetermined observance reaction. So this endotoxins is used for clot formation is interpreted as a positive result for the presence of endotoxin. If it is not, clot is formed, there is no endotoxins is present, and it will be exposed to temperature of 250 degrees Celsius for more than 30 minutes, and it will be bacterial. Endotoxins may cause fever, inflammation, and even septic shock. Thank you.